Polish cochineal, also known as Polish carmine scales, is a scale insect formerly used to produce a crimson dye of the same name, colloquially known as St. John's blood. The larvae of P.polonica are sessile parasites living on the roots of various herbs, especially those of the perennial knawel, growing on the sandy soils of Central Europe and other parts of Eurasia, before the development of aniline, alizarin, and other synthetic dyes. The insect was of great economic importance, although its use was in decline after the introduction of Mexican cochineal to Europe in the 16th century. Biology. Lifecycle. In mid-July, the female Polish cochineal lays approximately 600 to 700 eggs, encased with a white waxy ootica, in the ground. When the larvae hatch in late August or early September, they do not leave the egg case but remain inside until the end of winter. In late March or early April, the larvae emerge from the ground to feed for a short time on the low-growing leaves of the host plant before returning underground to feed on the plant's roots. At this point, the larvae undergo ecdysis, shedding their exoskeletons together with the legs and antennae, and they encyst by forming outer protective coatings within the root tissues. The cysts are small dark red or violet bubbles clustered on the host plant's roots. Female cysts are 3 to 4 mm in diameter. Males are half the size of the female counterparts and smaller in number, with only one male per 500 females. The cysts undergo ecdysis a number of times. When the male larva reaches the third instar developmental stage, it forms a delicate white cocoon and transforms into a pupa in early June. In late June or early July, females, which are neotenous and retain the larval form, re-emerge from the ground and slowly climb to the top of the host plant, where they wait until winged adult males, with characteristic plumes at the end of their abdomens, leave the cocoons and join them a few days later. Male imagines do not feed and die shortly after mating, while the female counterparts return underground to lay eggs. After oviposition, the female insects shrink and die. Host plants and geographic distribution. The Polish cochineal lives on herbaceous plants growing in sandy and arid, infertile soils. Its primary host plant is the perennial knawel, but it has also been known to feed on plants of 20 other genera, including mousea, hawkweed, bladder campion, velvet bent, carragana, smooth rupturewort, strawberry, and cinquefoil. The insect was once commonly found throughout the Palearctic and was recognized across Eurasia, from France and England to China, but it was mainly in Central Europe where it was common enough to make its industrial use economically viable. Excessive economic exploitation as well as the shrinking and degradation of its habitat have made the Polish cochineal a rare species. In 1994, it was included in the Ukrainian Red Book of Endangered Species. In Poland, where it was still common in the 1960s, there is insufficient data to determine its conservation status, and no protective measures are in place. History. Ancient Slavs developed a method of obtaining red dye from the larvae of the Polish cochineal. Despite the labor-intensive process of harvesting the cochineal and a relatively modest yield, the dye continued to be a highly sought-after commodity and a popular alternative to kermes throughout the Middle Ages until it was superseded by Mexican cochineal in the 16th century. Dye production similar to some other red dyes obtained from scale insects. The red coloring is derived from carminic acid with traces of kermesic acid. The Polish cochineal carminic acid content is approximately 0.6% of the insect's dried body weight. The insects were harvested shortly before the female larvae reached maturity, i.e. in late June, usually around St. John the Baptist's Day, hence the day's folk name, St. St. John's blood. The harvesting process involved uprooting the host plant and picking the female larvae, averaging approximately 10 insects from each plant. In Poland, including present-day Ukraine, and elsewhere in Europe, plantations were operated in order to deal with the high toll on the host plants. The larvae were killed with boiling water or vinegar and dried in the sun or in an oven, ground, and dissolved in sourdough or in light rye beer called kvass in order to remove fat. The extract could then be used for dyeing silk, wool, cotton, or linen. The dyeing process requires roughly 3 to 4 ounces of dye per pound of silk and 1 pound of dye to color almost 20 pounds of wool. Trade Polish cochineal was widely traded in Europe during the Middle Ages and the Renaissance. In the 15th and 16th centuries, along with grain, timber, and salt, it was one of Poland's chief exports, mainly to southern Germany and northern Italy as well as to France, England, the Ottoman Empire, and Armenia. In Poland, the cochineal trade was mostly monopolized by Jewish merchants, who bought the dye from peasants in Red Ruthenia and other regions of Poland and Lithuania. The merchants shipped the dye to major Polish cities such as Kraków, Gdansk, and Poznan. Further, the merchandise was exported to wholesalers in Breslau, Nuremberg, Frankfurt, Augsburg, Venice, and other destinations. The Polish cochineal trade was a lucrative business for the intermediaries. According to Marcin of Erzadau, 1 pound of Polish cochineal cost between 4 and 5 Venetian pounds. In terms of quantities, the trade reached its peak in the 1530s. In 1534, 1963 stones of the dye were sold in Poznan alone. The advent of cheaper Mexican cochineal led to an abrupt slump in the Polish cochineal trade, and the 1540s saw a steep decline in quantities of the red dye exported from Poland. In 1547, Polish cochineal disappeared from the Poznan Customs Registry. A Volhynian clerk noted in 1566 that the dye no longer paid in Gdansk. Perennial knawel plantations were replaced with cereal fields or pastures for raising cattle. Polish cochineal, which until then was mostly an export product, continued to be used locally by the peasants who collected it. It was employed not only for dyeing fabric but also as a vodka colorant, an ingredient in folk medicine, or even for decorative coloring of horses' tails. With the partitions of Poland at the end of the 18th century, vast markets in Russia and Central Asia opened to Polish cochineal, which became an export product again, this time to the east. In the 19th century, Bukhara, Uzbekistan, became the principal Polish cochineal trading center in Central Asia. From the dye was shipped to Kashgar in Xinjiang, and Kabul and Herat in Afghanistan. It is possible that the Polish dye was used to manufacture some of the famous Oriental rugs. Studies. The earliest known scientific study of the Polish cochineal is found in the Herbacts Polski by Marcin of Erzadau, where it was described as small red seeds that grow under plant roots, becoming ripe in April and from which a little bug emerges in June. The first scientific comments by non-Polish authors were written by Segerius and von Bernitz. In 1731, Johann Philipp Breen wrote Historia Naturalis Cocci Radicum Tinctoriae quod Polonicum vulgar audit, the first major treatise about the insect, including the results of his research on its physiology and life cycle. In 1934, Polish biologist Antoni Jakubski wrote Czerek Polski, a monograph taking into account both the insect's biology and historical role. Linguistics. The historical importance of the Polish cochineal is still reflected in most modern Slavic languages, where the words for the color red and for the month of June, both derived from the Proto-Slavic *crv, meaning a worm or larva, in the Czech language, as well as Old Bulgarian. This is true for both June and July, the 2 months when harvest of the insect's larvae was possible. In modern Polish, czerwek is a word for June, as well as for the Polish cochineal and its host plant, the perennial knawel.